my pleasure to introduce to you Professor Onur Mutlu. He's a distinguished professor at the Carnegie Mellon in the United States of America and at the ETH in Zurich, Switzerland. He's the most cited scientist in the field of memory systems and the third most cited scientist in the field of genomic analysis, with over 47,000 citations, according to the Google Scholar. His career is extremely prolific and versatile. He obtained his PhD and Master's of Science in Electrical and Computer Engineering from UT Texas in Austin. And also he has a Bachelor of Science degree in Computer Engineering and Psychology from the University of Michigan and Harvard. As a guest lecturer at the University of Montenegro, Professor already held a lecture today related to the field of hardware security and at the rectorate. And in addition to this, here at the Faculty of Medicine, he will be talking on the topic of analysis of genomic variation applications in pathology. Professor Mutlu, it is with great pleasure that I greet you on behalf of the academic staff of the University of Medicine, and I wish you a warm welcome.
Thank you. Thank you very much. And thank you for the invitation to give this talk. And thank you for the very kind introduction. I appreciate the opportunity to be here. This is my second time in Montenegro, and it's always great to be here, certainly. I should say that I'm completely an outsider to medicine. I have no medical degree. I'm not a doctor. I'm by training a computer engineer. So my perspective on these things will be the perspective of a computer engineer. If you ask me questions on if this disease is, how this disease is different from the other disease, I will be able to give you a zero answer. But I can give you an answer in terms of how you can accelerate some of these analyses potentially. And with my limited amount of knowledge, some bioinformatics also. So I went into bioinformatics. This is going to be a little bit of my story on going into bioinformatics also. I believe there's a whole lot of potential over here. And by enabling like this interdisciplinary research between medicine, biology, and computing, and even device design, as we will see, I think we can enable something much bigger going into the future, in my opinion, where there's a huge opportunity here. But that requires people to get together and actually talk to each other and actually do hard work to translate their languages to each other. This is one of the things that I found difficult, even within bioinformatics itself. Bioinformatics is a very large domain. It touches some medicine also, but it's difficult to actually communicate completely. By the way, I also figured out that I got this date wrong. That should be 9th of June, but I'm not going to change that right now because I don't want to mess up with this setting. I actually gave a similar talk at the UDG yesterday to a different crowd. So before I start, I guess I should ask, how many people have a medical background here? Okay, so quite a few. How many people have an engineering, like say electrical or engineering or computer science background? Okay, that's good to know also. How many people have some other background that's not medical? So, okay, so what is that, biology? Is it, okay, biology background. So that's great, actually. We have, this usually you don't get, actually. I'm really happy that we got all different, let's say, types of people. How many people don't fit in any of those categories? Okay, I'm curious what your background is. Mathematics, okay. Okay. Okay, wow, okay, amazing. It's good to certainly have people in those areas also because they actually have a lot of implications, I think. So, okay, In other words, this talk will be satisfying to no one because no one will be able to go into a lot of depth in their area, let's say, except maybe people who are doing bioinformatics and computer science similar to me. But hopefully you'll get something out of it. That's what I can promise, hopefully. Okay, so let's get started. So I think I'm going to give you my story, but I believe system design for bioinformatics is a critical problem. We need to design entire systems, computing systems, to do bioinformatics analyses. And clearly it has large scientific, medical, societal, personal implications. This talk is going to be about accelerating a key step in bioinformatics. Bioinformatics is a huge domain, and genome sequence analysis is a subdomain of it. And even in genome sequence analysis, I'm going to focus on a particular domain, which is read mapping. So specialization is very important, but we should never lose the big picture also. So I'm going to give you the big picture first, and then go and specialize a little bit. And I think the overall picture is that there are many bottlenecks that exist in accessing and manipulating huge amounts of genomic data during analysis. And there are clearly many analyses that we can do on genomic data, and also other types of data, other types of biological sequence data, and also other multimodal data that go into medical decision making. I'm going to look at essentially many recent ideas to accelerate read mapping. And this is going to be my personal journey since September 2006. This is after I got my PhD. I was working at Microsoft Research. I started the computer architecture group there. And I was basically interacting with people at University of Washington Genome Sciences Department who were doing some of these analyses early on, let's say. So we were basically brainstorming about how can we do these analyses better, let's say. So, okay, we were hiking at Mount Rainier, which is a beautiful mountain over there. Montenegro has a lot of beautiful mountains also. Mount Rainier is special for me because while we were hiking, we were actually brainstorming on what we can do in this based on our backgrounds and based on our limited vision and thinking at that time, right? Of course, you can always make your vision bigger. So basically, we were imagining an embedded device, something of this size, let's say, that you could give to a doctor or some person, right? It could be anyone that could analyze their or some patient's genome within a minute. That was our goal. So genome analysis was taking long. I'm going to give you some numbers. It's still taking long, I think. But we believe that if you could actually make genome analysis extremely efficient and extremely fast, it could have applications that could change people's lives, including doctors, patients. Clearly, there could be a critically ill patient, and you try to figure out what kind of drug should be applicable to him or her. And if you make the decision in an informed way using this device based on their genomic characteristics, hopefully you'll make the right decision fast enough and quickly so that you could save the patient in the best way, right? And these things happen clearly. I'm not a doctor, but I know stories from doctors that I've talked with that this could be useful to them. And you can certainly expand this to other analyses, right? We are focusing on genome analysis, but clearly there's other data that could be important, like protein analysis and also other data, but we're not going to focus on that. So clearly there are a lot of interesting questions, not just in medicine, but also fundamentally understanding evolution, right? Which of these DNAs does this DNA segment match with? That could be useful for phylogenetic trees of species and evolution, etc. I'm going to give you a little bit more. What is the likely genetic disposition of this patient to this particular drug that I'm considering? Or this collection of drugs, which one should I use, right? And what disease or condition might this particular DNA or RNA piece may be associated with? Some of these studies are done right now at very large scale. I'm going to mention them. They cost a lot today. They are done in data centers, for example. I'm going to also give you that scale, if you will. But we want to do it in a device that you can carry in your pocket and maybe do it even yourself, right? We're not there yet. Hopefully we'll get there. So basically, we need faster and scalable and efficient genome analysis. Clearly, this could be useful in what I said earlier, understanding genetic variation, species, evolution, predicting the presence or absence of microbes in a sample. And COVID-19 has taught us that understanding disease outbreaks, variations, and predicting them early enough so that you can actually do something about them could be useful. And developing other types of personalized medicine. And there are many, many other applications. So I believe there's a bright future for it. I'll give you the very high level first, and then we're going to progressively go into lower level details. But for example, you can have these nanopore devices today. These devices exist. At the time we were imagining, they didn't exist. People were working on it, but these were introduced in 2014 by Oxford Nanopore Technologies. So we were very happy that, oh, someone actually developed this embedded device that we were envisioning, except these devices are not what exactly we envisioned. They're very good at sequencing genomes, but they're terrible at analyzing data because they have absolutely no analysis power. but you can actually plug it into your phone and potentially analyze a little bit, not much. You can plug it into your laptop, analyze a little bit more, not much. You send the data to a data center, analyze using thousands of computers. Okay, you can do a lot over there, but that's not very efficient. Right? Okay, so our goal is to enable these devices to be extremely good data analyzers as well. So I should also say that I empathize with people who try to bridge this gap between, let's say, computation and biology. So we actually wrote a lot of articles to help, at least from our within our limited domain. IEEE Micro is read by a lot of people in our domain, for example. We try to educate them in terms of at least as much as we know about bioinformatics. So if you're interested in some of the basic articles, that was very difficult to find for me when I was actually going into this field, if you will. You can find some of these articles online. This is not the latest one. I'm going to mention this one. This is a bit more specialized. We were invited to write this article to this journal that's actually much more comprehensive, basically. Again, the focus is more computer science. If you're a medical person, you may actually learn something about computer science, hopefully. That'd be useful. And we actually write these. This is the latest one, but it's a short one. It talks about algorithm architecture co-design, which I will hopefully convey in a little bit. Okay, but let me give you the very high-level picture. So this is another genome sequencing machine. This is Illumina. It's one of the much more expensive machines. The other machine that I showed you is $1,000. This is maybe 2 or 3 orders of magnitude more in terms of price. In some cases, it could be actually even more depending on how many of these devices you buy, right? But basically, that device or this device, these are actually extremely good devices. These are sequencing machines. You put DNA or RNA into it, and they give you the sequence, which we will talk about more. They generate this genomic data, and they're very specialized for it. They're extremely fast, high throughput, low latency. There are some errors that happen, which we're going to talk about. But our analysis machines are actually extremely slow compared to them because they're not specialized. And also, there's another problem. Analysis is done far away from this device usually. Usually, you move the data from here to some data center, as I mentioned, or at least your laptop, which is usually not sufficient. So there's a lot of data movement that you cause from the specialized device, and you process the data in a non-specialized manner. And I think this is the fundamental problem I see today in building better analysis. We should really make these devices much more capable, specialized to genomic data, for example, in this particular case, and minimize the data movement that's happening. And I actually use this slide for my general computer architecture talks, because this picture is actually similar for many data generators and analyzers today. In the larger scale, we have a problem with data. So if you can replace this device with lots of video cameras. Video cameras are very specialized devices for video encoding, but we don't have very good devices for video analysis. And going forward, it's going to be a lot more important. And some of the techniques that we use in genomic analysis, like accelerating machine learning, could be useful for other domains like video as well. Okay, that's something else we work on, so I'm excited about it. But basically, if you look at computing perspective, you have algorithms to analyze data, and you have devices that you execute those algorithms on in the end. And there's a huge stack over here. You express your algorithms using programs, some system software runs on it, and then they get translated into hardware that actually does the work, transistors, basically. And we really need to understand this entire stack and specialize it for genomic data analysis, in our opinion. And I'm going to hopefully give you some examples. So, okay, this is an agenda which we'll get back to. So we're going to mainly talk about some algorithmic acceleration, but also hardware acceleration, but I'm going to motivate a lot of things first. So this is basic biology. Hopefully a lot of people know here about what a genome is. I will not go into a lot of detail clearly. Probably some of the other faculty over here can do a much better job than me than teaching this. But basically everybody has this, and we have a bunch of bases, A, C, T, G. And a genome is essentially a string of characters, but we're going to question that also in the end. And DNA clearly affects a lot of things. It enables transcription of RNAs, and then RNAs get translated into proteins, and they actually affect everything we do. Clearly, they don't have an absolute effect because there are other conditions that also affect things. But understanding the DNA can really help us understand basic things like why do we look the way we do, or some extremely important things like what kind of diseases we are vulnerable to and what should we do about it. Okay, and these are real things. This is actually, I like the story because this is Henrietta Lacks's genome, and if you've taken any genomic class, probably they've talked about it. This was taken from her out of her consent, without her consent, but this enabled a lot of good investigation into genomics. Okay, so this is my view, or let's say computer scientist view of genome. It's not this physical structure. It's more of a text structure, let's say. I don't know if this is stopped currently. Is it okay? Okay, as long as it's okay, I don't care, but it changed a little bit. But basically, genome is a sequence of these characters, which is bases in the end. And it's in human genome, it's 3.2 billion characters. There are smaller genomes, larger genomes. But in the end, what we're looking for is potentially some single base over here and what it is. It could change to some value in that alphabet. And that may indicate something really important, high blood pressure, for example, or a sequence of bases, or even a larger sequence of bases. The existence of them may indicate some propensity to autism, for example, to different kind of things. And this is the type of thing that we would like to do extremely fast. We don't want to wait 2 weeks or 2 days or even 2 days in these things. Okay, so this is a fun picture over here. This is to show that we're not very special in terms of the size of our genome, let's say. There are other things that have a lot more, let's say, base pairs in their genomes, and clearly there's viruses, etc. Okay, so let me go into more formally defining the problem. So we're going to be talking about DNA sequencing a lot. The goal in DNA sequencing is to first, initially, find the complete sequence of ACGTs in an organism's DNA or RNA. Pick whichever you want, right? The challenge is our technologies are good, but they're not good enough to do this completely, meaning there's no machine that takes DNA as input and gives the complete sequence as output. And we want the complete sequence so that we can do other analyses on top of it, especially important analyses that require the complete sequence. So all sequencing machines are actually choppers. They chop DNA into pieces and identify relatively small pieces, sometimes only 100 base pairs, sometimes 200, sometimes 300, sometimes 10,000 or 1 million, but not 3.2 billion. And they don't tell you how they fit together. So this immediately poses a computational challenge that takes a lot of the analysis time. So if you visualize what I just said, you have a large DNA molecule that looks like this. A machine chops it into small DNA fragments. And we're going to talk about the characteristics of those soon. These are called reads, essentially. These are reads that are essentially part of your genome. And current sequencing machines provide these small randomized fragments of the original DNA sequence. And you need to map them to a reference genome to really understand how they fit together first. And this actually takes the most time in most genomic analyses to begin with. There are, of course, other things. I liken this to a puzzle. So you have this puzzle that exists, and somebody breaks it into pieces. Pieces are not exactly the same size, and you have to reconstruct it. Now, if this was just this, that'd be good. It's not a perfect puzzle in the end, because when you're trying to construct it, you don't have a perfect reference also, because reference is different so my genome is different from yours. Everybody individual different. So you have to actually allow some differences between these different genomes when you're constructing this puzzle. So it's actually worse than a puzzle, let's say. I liken this to untangling yarn balls. If you have a cat, the cat may be playing with these yarn balls, and these may be different genomes. For example, when you do some microbiome analysis or metagenomic analysis, you actually take a sample, and you may have actually lots of different yarn balls, right? And then the machine chops them, and then your goal is to really figure out which piece of the cut yarn ball goes to which yarn ball. This is actually a very accurate representative of metagenomic analysis. When you're doing sampling for figuring out viruses that are lurking around in this room, for example, we do some sampling and we put it into a genome sequencer, we'll get these yarn balls in the end. And then the goal is to figure out what matches, what doesn't match. I'm going to talk about that also. But these are the choppers that we have today. Maybe the slide is not fully up to date, but some of them are extremely capable. Some of them are huge, as you can see. This one is huge over here. This one is very small. And there's a huge difference in terms of pricing also. You can buy the Oxford Nanopore machine for about $1,000 now. A lot of people can do the sequencing on their own. In fact, high school students do this. This is a motivational story, let's say. There are people who have studied their own fish's genome, not their own genome, but fish's genome. And high school students are actually enthusiastic about this. So it's pretty accessible today. And there are more. And all of them produce data with different properties, genomic data with different properties. Let's take a look at these properties after I show you this slide. So basically, the reason why we have an explosion of genomic data today is these sequencing machines have become extremely good. So if you, and essentially people are sequencing lots of genomes, and this is growing exponentially. And the reason is the cost is reducing. So if you look at this picture over here, of which I have a bigger picture soon. Yeah, okay, let's jump into this one. And maybe even this one, actually, cost per human genome. So with the introduction of some of these machines, high throughput sequencing technologies, the cost started dropping greatly. And that's the introduction of high throughput sequencing. This is the introduction of nanopore sequencing over here. So cost keeps reducing. And the interesting thing is the comparison to Moore's law over here. Moore's law basically dictates the cost of a transistor. So clearly the cost of a transistor is reducing exponentially, but the cost of sequencing is actually reducing even more. Now, this immediately says that if you don't do something about it, throwing more machines at the problem is not going to help you because the sequencers are becoming much more capable. That's why we're going to specialize in general. That's the motivation for this picture also, essentially. This machine is scaling much better in terms of cost and power compared to this machine, essentially. Okay, so let's talk about this data generator. So this is one of the data generators, and we've been working a lot with nanopore sequencing technology. It's an old paper by the standards of growth in bioinformatics, let's say. It was published in 2018. We analyzed, for example, programs that essentially do genome assembly as well as genome read mapping in this particular using this device. And you actually find a lot of bottlenecks. I'm going to talk about some of these bottlenecks, but essentially this is one of the data generators today. So I said that different machines produce data with different properties. So let's take a look at some of these properties. So some machines are extremely accurate in terms of the identification of ACTGs. Usually Illumina machines, PacBio machines are on this site. You get short reads. So this is a trade-off. Your accuracy of the fragment that you get is very high, 99.9%, but you get very short reads, meaning if you have a very large genome, the mapping task of figuring out where this read came from, that huge genome, is much more difficult compared to a long read. So it's not a perfect technology. It's very good at what it's doing. But then on the other hand, we have the nanopore sequencing technology, for example. You can get long reads. In fact, this has increased to more than 50K. You can get 1 million, 2 million bits today. But unfortunately, their accuracy is lower. You can see that it's around 90%, 95%. People are improving, but it's not as accurate as this technology. Now, why is this the case? You have to go into the underlying device technology, which is fascinating, but we don't have time to go over it. I want to draw an abstraction layer. Maybe I'll talk about nanopore sequencing a little bit later. But underlying devices are actually very, very interesting also. And we're working on some new nanopore devices, for example, to overcome the limitations of some of the existing nanopore devices collaboratively with biomedical informatics group at ETH. So I like the whole spectrum, very fascinating, including the device technology. So basically the downside is you may have long reads, which may make the mapping task easier, which may make it easier for you to figure out where this is coming from in the genome, but the accuracy is low. So actually you need to do something more on top of this. So usually what happens is because of the low accuracy, you don't sequence a genome once. You sequence the same genome 10 times, 20 times, 30 times, and build some confidence in terms of whether what you're seeing that's coming from the machine is actually correct. So why do we have these errors? Because the machines are not perfect. In nanopore sequencing, for example, you take DNA, DNA goes through a nanoscale hole. And in this nanoscale hole, while the DNA is passing, you measure the electrical current it causes. And electrical current is different based on the bases that are passing through this nanoscale hole. So this is not a perfect process. So the device is imperfect. So we cover up for the imperfections by doing sequencing many, many times and building some confidence as to so that we can actually correct for these. So this makes it actually even more costly, right? Sequencing one genome once and trying to map is costly. Now imagine doing it 30 times to get accurate results. That increases the cost by 30x. Okay, so that's the need for better devices and also better analysis. Okay, so basically, I think the takeaway over here is we have these great sequencing devices, regardless of their faults. Unfortunately, scientific discovery and medical advancements are bottlenecked by the steps that are taken from the devices and all the way to where it goes, basically, to the scientist or the doctor. And that's why I'm going to talk about some of these steps over here. You map the reads first, for example, to understand where they're coming from. And then you ask the question, is this actually an interesting variant that I should examine? Is this a single nucleotide polymorphism, for example, that we will see that's relevant for the patient? Or maybe we discover something based on this particular variant that I've never observed before, right, in any other genome. So there are, of course, other analyses over here. So this read mapping process is interesting. We recently written a survey article. If you actually want to go into this area, this is a very small piece of the entire bioinformatics area, but even that area has a lot of work in it, like 40 years of work. We wrote 111 pages together with lots of people who are working on read mapping. So if you're interested, you can take a look at it. So let me go into why do we care before we go into a little bit more detail. So I think we care in several different things. So if for example, I give you a bunch of sequences, multiple sequence alignment enables us to tell in what parts they're different, in what parts they're similar. This could be very useful for, again, scientific analysis, but also disease analysis. I like this picture over here where different bacteria are actually aligned, let's say, in terms of their genomes, and you can figure out in what parts they're different or similar. So you can actually understand the evolution much better this way, which is scientifically nice. Now, this is also interesting. Many people may not realize that we are much more similar to each other than we think, let's say. As you can see, these people may be completely opposed to each other, but they're very similar to each other genetically in the end. And of course, the funny part is maybe we're very close to bananas than we think, as you can see genomically. But I think the interesting thing is here, this number, right? So this is a part of the difficulty. We have very similar genomes to each other. Sometimes we really want to identify what's different. And if you have a huge genome and you are trying to identify what's different, that complicates the problem. If the differences were huge, that's easier to identify. You can actually use a lot of algorithmic techniques that are approximate that way. But if you want to focus on something that's really specific, that's different, you have to be accurate. And that accuracy is very important. So I can easily identify human versus banana genome with some quick searches algorithmically, but this is much more difficult to identify. Okay, so hopefully that gives you an example. And this is an example where you really want to identify particular changes. So for example, blood pressure has been shown to be correlated with particular SNPs, single nucleotide polymorphism, particular parts in the genome where a single base is modified, basically. This is done through analyses that I will show you at the high level in the next slide. But this is one of the questions that you could ask, for example. Am I genetically inclined to have high blood pressure? And that requires analysis and figuring out those SNPs. And if you actually have a variation in that SNP compared to a healthy person, let's say, or a person that is statistically determined to not have to have a particular value in that position, that's not correlated with high blood pressure, right? So that's a more accurate way of thinking about it, because all of these are statistical studies. You sequence lots of genomes. You try to figure out whether a particular piece of the genome correlates with high blood pressure versus low blood pressure. Everything is statistical in the end, right? And this is one example. These are called genome-wide association studies. A lot of people, biostatisticians, for example, do this a lot. You may be doing this a lot in some research, I don't know. But essentially, this enables detection of genetic variants associated with phenotypes using two groups of people. In this case, we look at people without heart disease, people with heart disease. And you want to identify these pieces of the genome that occur in a particular way in people with heart disease, but do not occur in that same way in people without heart disease. That's the idea. Of course, to do this analysis, you also use a lot of machine learning type techniques, statistic techniques, but also machine learning type techniques today, which is not what I'm going to talk about today. That's also fascinating, and we want to accelerate that also. And we actually are building accelerators for that. A lot of machine learning accelerators, a lot of clustering accelerators, data clustering accelerators are actually quite applicable to this, and I find those fascinating. But we don't have time to talk about all possible acceleration studies, but there's a lot of applications in this sort of acceleration also. And again, I'm going to talk to you about what I know. I don't know a lot of things in medical studies, etc. There could be something that you may say, oh, this problem actually I have. It could be solved with better acceleration, right? If you have that, I'd love to learn about it. Okay, so clearly this could be useful for other things, and people show, I have links over here that talk about, for example, you can find these SNPs that correlate with different diseases in open databases. You can actually find these online. They tell you, these are knowns. Knowns are nice. Even known analyses take a long time, but unknowns are actually even worse. So if we don't know something, it requires even more analysis to understand that. Okay, so there are also a lot of other studies that talk about these, let's say, different conditions. They're associated with actually particular chromosomes. As you can see, there's a special chromosome. And you can actually find out some of these things with specialized analyses that require read mapping in the end, that require what I just mentioned. I like this one over here because I think this is kind of in line with what we had envisioned in 2006-07, right? Basically, there are a bunch of papers that talk about the utility of genome sequencing or exome sequencing, etc. It's a version of genome sequencing, I would say, essentially to understand the vulnerability of critically ill infants in this particular case. And these people report that because of genome sequencing, they were able to treat this critically ill infant. But they also provide numbers, for example. For example, if you actually, you need to wait 5 days in some cases to get this analysis. If you want it faster, you need to pay a lot more to get this analysis done. So clearly not everybody can afford it. Our goal is to actually design these systems such that everybody can afford it and can be very quick. We don't want 2 days. We want 1 minute, basically, if possible. So it requires, we're still not there, but the goal is actually challenging also, I should say. Okay, so if you're interested in this sort of thing, let's say, this is a very nice paper that was published by these people on Nature Reviews Genetics that talk about applications of structural variation. These variations in genome is called structural variation in general. It could be single base pair variation, it could be large variations, it could be copy number, etc. They're in general structural variations in the genome, and they talk about a lot of interesting things about how this could be applicable from their perspective, of course. Okay, so clearly you can also do other things. For example, you can identify a bunch of short sequences, and you can actually have, let's say you have randomly some short sequences. Can you identify who do they belong to? And this brings us to some interesting studies like microbiome profiling. You can actually sample things over there in that city and figure out what's going on. You may actually ask interesting questions. Are there some interesting things lurking here that I should be concerned about, right? And people did this actually in this beautiful paper. They did this study in New York City, and they sampled, for example, in the subway in the New York City, a lot of different things. And they did some study based on what they know at that time. And they found out a lot of bacteria, a lot of known things, but 48% unknown organisms, right? So do we actually know these algorithms? Do we not know these organisms? Unknown is a very general umbrella term over here. So what are these? It'd be good to identify these. So if you actually identify these early, maybe you could actually find COVID-19 or something like that, right? That's the hope, basically. But this requires a lot of analysis also. And read mapping, again, sequencing the genome or sequencing the RNA is actually the first step in all of these. Okay, there are a bunch of other papers that are actually trying to do even more. This paper, for example, this was published recently in 2 years ago in the Cell, and they basically trying to construct a metagenomic map of urban microbiomes. Essentially, they're doing this in a much larger scale than New York City, I should say. And the data is growing, actually. So some people project that at this point, we have a zettabyte of genomic data, 2025, we're almost there. I don't know if this is true, but this is the projection. This part is definitely true over here. So going forward, I think data is exploding. So analysis of it is actually becoming much more difficult as well. And also, I should also mention that these devices have been extremely instrumental in understanding COVID-19 and its variants. This is an article from 2020, early 2020, where a bunch of these nanopore sequences were shipped to China, where the pandemic was the fastest growing at that time, to really understand the variants. And these nanopore sequences actually enabled a lot of understanding of the variants and development of also vaccines for COVID-19. I'm putting this example because a lot of you lived through it probably and understand it. And there are papers written about it, which I'm not going to talk about. But I should also say that this is not new. People were doing this in Africa for Ebola, for example, in 2015, 16, 14, 10s. And this is one paper where they talk about their experience, but you can see the infrastructure that they have, right? It's not very sophisticated. We want to make this extremely efficient going into the future. Okay, so hopefully I've convinced you that we need faster, scalable, and more efficient genome analysis. And there are many applications here, some of which we can imagine, some of which may not have been imagined by me or my co-authors, right? Now let's jump into a little bit more detail. If there are questions, I'm happy to take them at this time also. We don't need to wait until the end. So feel free to interact. If not, I can keep going. And please stop me if I'm going too long. I tend to do that. Okay, so now let's jump into this problem of read mapping. Basically, we have the sequence and we want to analyze it. What's the first step? And this may be a bit boring for people who are not excited about, let's say, computer science, but hopefully it'll be instructive in the end. So basically, we're bottlenecked by read mapping in our machine. So there are a lot of measurements that people do. Sequencing machines are much, much faster than analysis machines. So these are some of the papers that I mentioned over here. So this paper, for example, reports these numbers. You can actually do 40x human whole genomes at that time. This is 2017. You can actually do this sequencing in about 2 days, which is not bad. 30x covers means that every genome, every DNA is sequenced 30 times so that you can actually build confidence, right? But on a very sophisticated processor, you can analyze one human genome using 32 CPRs. Basically, it's much slower. Okay, so this is basically the picture. Now I put some data into the picture. with some caveats, of course. It depends on what kind of analysis that you do also, right, in the end. You may actually do some analysis that takes even more, like we saw in the critically ill patients, right, earlier. Okay, so the problem I'm going to focus on mostly, but not fully, is we want to construct the entire genome from many sequence reads. So you immediately have a problem actually after this machine. We have many short DNA fragments or reads. We start with mapping them to a known reference genome with some differences allowed. So DNA logically looks like this, physically looks like this. The chopper goes and chops this DNA into what's called reads. And then somebody with a lot of effort constructed this reference genome. So human genome took, I don't know, 10, 15 more years to construct, actually. It's still being constructed. It's not fully constructed yet. We still don't understand all of it like fully. But somebody constructed the reference genome mostly. And our goal is to actually, this is my genome. The human genome is someone else's genome, someone else who donated that genome, right? I'm going to map these reads to this reference genome to have an idea of what my full genome looks like. That's the idea. Of course, my genome is not going to match perfectly that person's genome, that reference genome. As a result, that's why we have these some differences allowed. That's one of the reasons. So we have variations, and those variations are the interesting parts to detect, actually. How do I not match this other person? And is that indicative of something good or something bad, essentially, based on the analysis that I'm doing? And then there's another complication. We want to allow these differences because the machine is not perfect also. In some cases, the machine tells you it's an A, but it's actually a G. And this sort of errors also happen. So you really need to detect these variations and need to do it accurately. Okay. So this is challenging because the reference genome can be 3.2 billion bases, whereas small fragments, small reads could be 300 bases. So metagenomic analysis makes it even more complicated. So now we have reads from unknown donors at sequencing time. You try to map them to many known reference genomes or a reference database, if you will. So if you sample this place, you'll get a bunch of genetic material recovered from the environment. And then these get chopped into pieces. You normally don't have colors, actually. This is just to visualize. You don't get actually any colors. You just ACTG, right? All of them are ACTGs, for example. And then the goal of metagenomic analysis is to figure out in a huge reference database, what is this basically? What am I looking at? The purples map mostly to this genome. So maybe it's that, right? But some of it maps to somewhere else also, right? Because these are small reads. So you actually get false positive mappings in this case also, right? I don't know if you kept up to date with the story of COVID analysis, right? This Wuhan market, people analyze, basically people do metagenomic analysis to try to understand what's there in the Wuhan market at that time. So there's this controversy about raccoon dog you may have heard about, which is a fascinating controversy, I think, which I will not go into. But essentially, I believe, based on the data that I've seen, that raccoon dog is a false positive. It seems like there's some sample that is taken that matches this raccoon dog DNA or, yeah, okay. Anyway, I will not go into this in more detail, but basically not carefully doing these analyses accurately may lead to potentially false conclusions that may have huge political implications also, as we see in COVID-19, for example. Okay, but hopefully that gives you an idea about what this metagenomic analysis could be useful for. It can actually help you trace back what was there in the market at that point in time, assuming you do it accurately, and how it correlates with the RNA of the COVID genome. Okay, now let me go into a more computer science perspective. Computer science doesn't work on those squiggles. It works using these text. I'm going to question that later on also, so I keep that in mind. So usually we represent reference genomes using files. So the sequencing machine generates eventually these files. It's a huge text file, basically. You can find a lot of them online. A lot of this is published. And when you sequence someone's reads, the sequencing machine also generates these read files. So a reference genome is stored somewhere. Sorry, sequencing machine doesn't generate that. You actually constructed it. When you sequence your own genome, for example, this is what you get. This is a piece of what you get. This is a small read, for example, short read. Just focus on the ACTGs. Basically, our task is to figure out that these three pieces over here may be mapping here. I don't want to figure out that this other blue piece, this may be a false positive, because these three pieces are consecutive and these three pieces are consecutive. So probably this consecutive read is coming from here. Okay. So it's easier to set than done, right? Because you don't have these colors, as I said. So how do you do this? So now it's going to become more interesting to computer science and mathematicians, maybe. Basically, you need to do some sort of base by base comparison. You have these texts, references a text, reads the text, you need to do comparison. This comparison is not easy. This is called edit distance computation, and there are a lot of fundamental algorithms that were developed early in the 1980s. And actually, there's some breakthrough that are happening right now in the field also to get rid of the useless computation that these algorithms do, which is interesting. So essentially, what are we trying to do? We're trying to approximately match these two different texts, and we're trying to find the minimum number of edits, insertions, deletions, or substitutions that are needed to make the read exactly match or align with the reference segment. This is called edit distance. This is a very fundamental definition. I'm going to give you an example of it. So pick two of your favorite countries. It doesn't have to be these two. Essentially, if you want to align Netherlands to Switzerland, you need to realize that the first two characters don't match, right? So there are mismatches over here. There's a deletion in Netherlands that compared to Switzerland, right? And then there's a match at T over here, and there's a mismatch over here, and there's a sequence of matches over here, Erlands, and then there's an insertion in Netherlands that doesn't exist in Switzerland, right? So either it could be the reference over here, the other could be the read. Assume that Netherlands is the reference, Switzerland is the read. Basically, the read has an edit distance of 5 characters compared to the reference in this particular case. Now, do we want to detect 5 in this particular case? Then the question is, what kind of variations are you trying to detect? So ideally, you should be able to have an algorithm and system design that enables you to say, oh, I want to detect differences that are up to 10% between the strings, or 30%, or maybe 50%, right? As that number gets higher, this is called the edit distance threshold or error threshold, it becomes much more expensive to do this algorithm until that number becomes 100 or close to 100. Because 100% exact match detection is very easy. It's simple comparison of every character, right? If you have a mismatch somewhere, you're wrong. So basically, there's an area that's actually very tough to do, tough to scale, because the errors are relatively high in the read compared to the reference. But we would like to be able to enable that number, because when I ask, for example, people who are doing bioinformatics analysis, what should be that number? Everybody gives a different number, and a lot of people say, I don't know. So if you're building tools that these people are trying to use to detect variations, I don't know is not a great answer. So what do? Basically, you design algorithms that enables people to input whatever they feel like they're inputting at that point based on their current knowledge, right? So that number is important, as you can see. So that's what we are trying to enable, essentially. So there are many challenges over here. One is you need to find the many mappings of each read. A short read may map to many locations, especially with high throughput sequencing technologies. How do we find all of the mappings efficiently so that we can discover what is the right position in the reference genome? This is complicated by the variances and errors in each read, because each individual is different. Subject's DNA may slightly differ from the reference, and also the machines have errors. So how can we efficiently map each read with up to E errors present? As E increases, this becomes more complicated until E gets very close to 100%, which is not that interesting potentially, right? 100%. And then you need to map each read very fast. Performance is important. I already mentioned this. So how can we design a much higher performance read mapper? So let me also give you a little bit more algorithmic and mathematical insight into why this is difficult. So read alignment is usually done using quadratic time dynamic programming algorithms. So this is the reference string, this is the read string. You use something called dynamic programming in computer science. How many people know about dynamic programming here? Okay, so the computer science are visible here. I will not go into a background of dynamic programming because that's not, let's say, so interesting unless you want to optimize the algorithms. But I just want to give you the complexity of this. So you really need to traverse this entire matrix to figure out the distance, and then you need to back traverse it also, essentially. So data dependencies limit the computation parallelism, and entire matrix needs to be computed, even though strings may be dissimilar. Recent advancements in dynamic programming or this sort of matching actually tackled this one. Basically, how can I make sure I don't compute the entire thing so that I can maximize the efficiency? So there are actually some algorithms like the wavefront algorithm that are developed that are actually quite interesting, which we're actually also working on. So, okay, Netherlands versus Switzerland. Basically, you need to compare each positions. Eventually, you get to 5. So basically, these show the edit distances of different prefixes of these different, let's say, characters. Eventually, this number is what you're looking for, 5. So you need to compute the entire matrix in this particular case because you have to see everything over here, but you may not, you may actually prune the matrix over here. So essentially, it's complicated, even with small strings. Imagine now the strings are 10,000 long or 1 million long. That becomes even more difficult. Small strings are difficult, but larger strings become even more difficult. Okay, I will not bore you. Basically, this is what this table is trying to do. It's trying to compare all possible prefixes of Switzerland to Netherlands, if you will, and find the differences. If you didn't understand that, don't worry. There a lot of people, when they're taught dynamic programming, immediately don't understand it. I didn't understand initially, for example. I had to read it 10 times probably to fully understand it in the end. Okay, so basically this is costly, and people write theoretical computer scientists write articles like this, basically saying that this cannot be computed in subquadratic time, so it's actually extremely expensive as the strings grow. This is a beautiful paper if you're a computer scientist or a mathematician. And that's why actually people have been working on this for 40 years, how to actually make this efficient. Okay, even though there's been a lot of work, today, actually, whenever you do the read mapping, 60% of the read mapper's time is still spent on sequence alignment. So I will not go into a lot of details over here because we don't have a whole lot of time. But now let me talk about acceleration a little bit. How do we accelerate these things and accelerate things in generally with a focus on read mapping? So a read mapper actually today consists of a lot of sophisticated structures. So reference genome, maybe I should give you the complexity of the problem actually. One, let's say, not so great way of doing the comparison. You have this reference genome, 3.2 billion base pair, and you have this 300 base pair read. One not great way of reducing doing the mapping is you take the 300 base pair read and compare it to every possible position in the 3.2 billion base pair genome, do the edit distance computation for every single position. Now that's extremely expensive. That's how past sequence alignment analysis methods used to work, like BLAST you may have heard of. It's extremely slow. It's extremely popular at that time, but it was extremely slow because of this reason. So modern mappers are actually much more database-like. So what they do is basically they index the reference genome, they chop it into pieces, and using this index, you try to figure out, oh, is this particular read that I just got from the machine, where is it located in the reference genome? So you have an index that tells you which locations. Even that's costly, but much less costly than computing edit distance for the entire reference genome for every single read. So hopefully this gives you an idea of the complexity of the problem. So even with the indexing, we're costly. So I'm going to talk about some of these things over here. Basically, you want to avoid the sequence alignment as much as possible, this costly edit distance comparison as much as possible because it's extremely costly. So we're going to see some heuristics techniques to accelerate this. And again, for more detail, I can refer you to some papers. And we actually also have a genomics course that we've been developing over the course of several years. You can attend this online. It's all live streamed on YouTube as all of our courses, but maybe you'll find some interesting material over here. A lot of the material are that they span actually computer science and also some biology. So if you're interested, you can find something interesting. We talk about genome assembly, which I'm not going to talk about today, for example. So maybe I'll mention that because it's interesting than going into a lot of detail in a particular topic. But genome assembly is the idea of what if you don't have the reference genome? How do you construct it, essentially? This is the first time you actually sequence some species. Humans used to be like that some number of years ago. And how do you construct the reference genome? This requires a lot of work, actually. And there's a lot of computation as well as biology that goes into it. Again, I will leave it as a teaser if you're interested. But it's also a fascinating and much more difficult problem than what I'm talking about. But I believe that's interesting, but it's not as critical as what we're talking about in the sense that once you've constructed the reference genome, that's good. You can always improve its accuracy, etc. But once you've constructed it, a lot of the analyses that you do is actually querying the reference genome, which is really trying to map the reads that are coming from whatever species to that reference genome or to many reference genomes. So assembly is normally a one-time process. Maybe down 20 years, 30 years down the road, quickly assembling genomes actually is very interesting, but we're not there yet, I think. Okay, so we actually also hold these workshops, held this workshop at RECOM, which is the premier computational biology conference, which was held in Istanbul in April. And we talk about acceleration. So if people are interested, they can learn more about it. Okay, now let me jump into a little bit more detail, which is hardware acceleration. And I'm going to give you some basic ideas. I'm going to introduce some technologies. FPGAs are field programmable gate arrays. These are reconfigurable arrays. They're not general purpose computers. Today we have CPUs and then GPUs, graphics processing units. GPUs are much more parallel. And then we have these reconfigurable architectures that are a little bit more, let's say, expensive, but you can reconfigure what the hardware does. Essentially, at the circuit level, you can configure it. And we've been working with these a lot to accelerate read mapping, as you can see what you've talked about. But let me give you the key idea that we've been looking at. Basically, the key idea is eliminate useless computation as much as possible, eliminate data movement as much as possible. I think I've given you all the secret sauce of accelerating genomics, but how do you do that becomes problem. Basically, filter fast before you do the expensive computation. In this particular case, minimize edit distance computations. Focus your effort into what's really important. I think in other ways you can think about it that way. So if you have sequence trees, some of them are going to be similar to the reference. Some of them are going to be too dissimilar from the reference. So if we can identify the red part over here, too dissimilar from the reference very, very quickly, we don't need to do any costly computation on them. And similar to the reference ones are the really interesting ones. This is where we need to really carefully ask the question, oh, is this a variation? Or what is the percentage variation here? Is there an error in the machine, etc.? So we want to focus our efforts basically, and that's been actually our effort to do. And I think this idea is generally applicable to any type of genomic analyses and any type of analysis in the end. Meaning, if you actually know what you're looking for, try to get rid of all of the other computation that you don't need. I don't think this is done. For example, early bioinformatics tools, as I mentioned, they were trying to search this read in every possible place in the entire reference genome. That's why they were slow. But people said, oh, let's build an index so that we don't search places that we know it's not going to match, even with, let's say, 15% error difference, for example. So I think this is very important in building any tool going into the future. It's not limited to bioinformatics, in my opinion. Okay, so actually, when we first started this area, we built a read mapper. I don't show this picture over here. We worked with the University of Washington Genome Sciences Department. Those folks were actually very esteemed in the sense that they worked in the sequencing of the initial human genome, which was a huge project, but they did their part in it, let's say. So they were actually very experienced. It's always good to collaborate with people who have experience, let me put it that way. And they had also designed a read mapper, which I personally accelerated, actually, made it work very fast using some compression mechanisms, etc. And then we said, basically, can we accelerate it even more? So we applied this approach to software first, completely using software methods. We tried to get rid of computation. I'm not going to talk about that. I'm going to talk about something here, because this introduced an interesting metric. How many people know about Hamming distance here? Again, computer scientists may know about it, maybe. Okay, Hamming distance, computer scientists knows about it over there. So let me give you the idea. So these are two strings. They're equal strings, right? Hamming distance basically computes the differences in terms of number of characters. And essentially, in computer science terms, you XOR, exclusive OR, each of these characters, and you find out that all of them match each other, right? In this particular case. So you get 0 mismatches. Now, if I delete a character over here, it looks like a terrible name, but this is what happens in genetics, right? Essentially, we have one edit, one deletion. I know that the edit distance is 1. Hamming distance is easy to compute. That's why I started with this. Basically, you XOR, you do exclusive OR of each character independently, completely independent of each other. You don't need to build a matrix or anything. Now, if you actually do this edit, Hamming distance doesn't work anymore, right? The Hamming distance between these two strings, assuming you padded this one with a 0 over here or whatever, is 5. Maybe basically you get 5 mismatches. Okay, so our insight to accelerate this sort of matching was to shift the second string to the right once. Basically, we compare the original string, which may be the reference, to the read that we've got. And in order to find the distance between these, we compare the reference to many different versions of the string, shifted left and shifted right. That enables us to cancel the effect of deletions as well as insertions. So let's take a look at what happens. So when you compare without the shifted version over here, you get 3 matches, 5 mismatches. Now we shift this to the right. Now we're getting something interesting, right? So you get actually matches in bull over here. So if you actually, the entire idea is doing this shifting many, many times and then comparing, and eventually, basically each comparison is an XOR. And eventually, well, this is the XOR results. And then if you add these two results, essentially you get one mismatch over here. So you count the number of ones. If it's greater than a threshold, then you say, oh, this is not going to match, probably. So I should not do any complex computation on this anymore. So hopefully you get the basic idea. With very simple computations like this that are simple, approximate, not perfect, imperfect, you can actually get an idea of whether two strings are dissimilar enough from each other that you should discard them from further analyses. And I like this because this actually accelerates things a lot. We showed that even on CPUs with this accelerates things by 3x or 4x, but it's actually nicely accelerated with FPGAs. So if you actually go back to the matrix, you don't have that dependencies anymore. Everything is independent right now. This is the good part. And these are, if you look at the matrix terms, these are the 2 deletion masks over here, and then the 1 deletion mask and 2 insertion masks. Okay, I will not talk about them more over here. But basically, you can build an FPGA, field programmable gate array, and then configure the circuits to do this computation extremely efficiently, because it's very bitwise operations. It's very independent. And as a result, this is the FPGA circuit, which at least the layout, it actually doesn't occupy a lot of area also, as you can see in the hardware. And what we get is essentially a 10x speed up in read mapping. So every other analysis that comes after this gets accelerated, of course, right? And also there are some other properties that I'm not going to talk about. And this is all freely available online. You can use it if you're interested, but we've superseded it, if you will. So, okay, let me give you the vision also. So we greatly speed up read mapping. And we actually, when we were doing this work, we said that this could be integrated with the sequencer itself. So sequencing machine can have an FPGA inside such that it can do some sort of initial filtering of what's interesting. Of course, hopefully based on some bioinformatics guidance, right, as to what's important and what's not important. So we can enable real-time filtering while sequencing, and this paves the way for on-device genome analysis. And of course, we're not there yet, but interestingly, Illumina, for example, has a platform that's very similar today. It's called the Dragon platform. They have FPGAs inside. They don't talk a whole lot about what they do exactly. I think if you actually pay them enough money, they will be able to talk to you, perhaps, and tell you how you can configure it, which is the good part, I guess, of working with them. But essentially, you can do something similar in an Illumina machine today. So that's the good part. So things are actually moving in the right direction, in my opinion. Okay, so basically, this is an example of what I mean by algorithm architecture co-design, right? This is, we want to get rid of useless computation. So we design algorithms and metrics such that they are a very good fit for accelerating inside the hardware. So hopefully you got some idea over here. Now, there are a lot of other works that I'm not going to talk about because we don't have a lot of time, but you can actually accelerate things in real hardware. I will mention one thing over here, depending on how much time we have. This is the latest filter that we have developed. All of these are published in bioinformatics, as you can see. Essentially, you can actually do much better than what I just said. I'm going to skip this. But basically, this is more algorithmic. So if you want to try to figure out if two strings are dissimilar to each other, you can convert the algorithmic problem from string matching to something that we actually know very well in VLSI design, in hardware design. It's called the single net routing problem. So you have the snake over here. You want to route an input to the output, and you want to go through a minimum number of obstacles. You can think of the obstacles as differences in the two strings. And if you actually express the problem this way, you can have a much faster algorithm accordingly. Again, I don't want to go into a whole lot of detail, but you can see the speed ups over here. It's 1 to 2 orders of magnitude speed up compared to existing CPU software that is used in bioinformatics today. I should probably ask how much time we have depending on the detail. Are we have time or? Okay. Okay, I'll let you stop me if we're running out of time. So I will not go into detail again, but I will mention that these are the matrices that I showed you earlier, shown in a row and column format as opposed to other format. Basically, these are the differences between these two different strings over here. One of them shifted left to the right, left once, twice, 3 times, and the other one also shifted right once, twice, 3 times. And these are the Hamming masks that you have that I showed you earlier. Now, Sneaky Snake, what it does is basically it says, oh, ones are not matching, zeros are matching. So ones are black, they're obstacles. Zeros are not black. They're basically the long sequence of matching strings. So you start from the entrance, and then you race to the exit, if you will. I like algorithms, as you can see. Basically, the snake figures out that initially it can go 4 distances quickly until it sees an obstacle. So you do that instead of going through, for example, here. You get to the obstacle very quickly. And then you reduce the error count that you can tolerate. And then you figure out that, oh, you can go 4 more distances over here. And then you reduce the error count that you can tolerate. And then you can pick either one of these ones over here. And then you reach the exit. Basically, in this particular case, we have figured out that in less than 3 errors, or exactly 3 errors, you can actually find matches between these two strings. So you should not eliminate this from further consideration. If your error count became negative, for example, you could eliminate it with some confidence. That's the idea. And actually, there's a lot of optimization you can do. You can build this on the fly. So in the end, you can actually speed up existing read mappers significantly, according to our results. And other people actually replicated these results, which is great. But of course, it takes time to actually get this adopted in bioinformatics analyses because things move relatively slowly in general when you change something in software. Okay, so I will also mention that we work together with other companies like Intel. Intel actually manufactures CPUs, as you may know, and we look at approximate string matching acceleration frameworks. Again, I'm not going to bore you with the details over here, but here we want to find algorithms that are very good matches so that you can accelerate them. And here we want to be a lot more applicable. So we actually have new algorithms over here. Okay, and this is the accelerator design if you're interested. There are a lot of interesting things, the hardware as well as algorithm design. I'm going to skip this. But what I will mention over here is some of these accelerators resemble machine learning accelerators a lot. Today, people are building machine learning accelerators. For example, Google, Amazon, Microsoft, everybody has their own machine learning accelerator. And their goal is to accelerate machine learning. This is useful for medical and bioinformatics analysis on its own. But you can also repurpose some of these accelerators to accelerate some specific things that we are doing also. So there's some synergy in terms of the broader acceleration of applications domain as well. Okay, so I think this will be boring to people, so I'm going to skip this one. If these slides will be available, and I think I'm going to send the recording also to people so you can find them. So the interesting thing is you can integrate something like this on an Intel CPU, and it will occupy only 1% of the area additionally, and also power. So that's one of our goals also. Can we integrate them into the general purpose CPUs that are in everybody's pockets with very little cost so that people can do these analyses much more efficiently? And what is much more efficiently? Essentially, if you looked at many different applications of this GenASM algorithm, let's say, you can accelerate read alignment, you can accelerate speed alignment filtering, it is distance calculation. And we essentially see significant speed up. Some of these are tools that are used very heavily in the field, Minimap2 or BWMM, for example. And you see that the speed ups are more than 2 orders of magnitude, and power consumption is also lower. So we're getting in the right direction, if you will, making things faster as well as more energy efficient. Okay, if you want to learn more, there's more over here. I should also say that that was, let's say, this was published in 2020. I have to look over here. But more recently, one of my other PhD students figured out how to make things even faster. So there's a lot of movement that's happening. And again, I will not go into the details over here, but you can actually be even faster than the speed up that we have shown earlier with some careful thinking of the algorithms. Okay, and you can find this online. This is the state of the art currently. Okay, let me also give you one more thing. So this problem is actually going to get much worse in the sense that, as I said, reference genome is a linear genome today. Actually, it's changing. It's not a linear genome because what we talked about, right? This is someone's genome. It doesn't represent the entire humanity. This is just that person's genome. People are thinking about how can we incorporate variations in the representation of the reference genome so that we can actually identify things much better. For example, a European, whatever you call European, right? Let's pick European as a continent, general continent, which is diverse in itself. But a reference genome that's constructed for Europe is very different from a reference genome that's constructed for Africa. And once you know about information about the different vulnerabilities and different environmental conditions, you really want to use a reference genome that you, let's say, belong to with some definition of belong to, right? So people are actually constructing reference genomes right now, not as a linear sequence, but as a graph, meaning they're representing the reference genome variations using some graph structure. And this makes analysis even more complicated because now instead of comparing two sequences like what we were doing earlier, you need to compare the reads, which is a sequence in the end. It's not a graph at that point. That could also potentially be a graph, but maybe 10 years down the road. But you have a sequence and you're trying to compare whether it matches a graph. And that's even more challenging computationally. So as reference genomes become more complicated and more comprehensive and more accurate from a medical and bioinformatics perspective, because you really want to try to personalize the reference genome representation to the groups, the computational analyses become more expensive also. And we show that in this paper. And we basically try to accelerate it. Again, I will not bore you into details over here. And this process is even further because you need to construct the reference genome indexes, etc. And in the end, the hardware also becomes more complicated. But if you can accelerate it, you can actually get a lot more speed ups compared to pure software tools that are state of the art today. So there are software tools that are not there yet. So you can actually get a lot of speed up. Okay, so hopefully that gives you an idea of the speed ups. And now, I will also mention some maybe future directions. So far I've said, there's a lot more we can talk about depending on the time, but so far I've kind of said reference genome is text. We're trying to deconstruct that also. In the end, the genome is not text. What you're doing is actually, this is a nanopore, nanoscale hole, as I said. DNA is passing through it, and you're measuring a signal. That signal is current. So if you look at this picture, it's more representative, if you will. This is some sort of nanopore device, graphene. DNA strand goes through it, and you actually measure the electrical current perturbations that happen. And you try to match them to ACGTs, essentially. There's nothing that says we should convert that to text. Why don't we just directly operate on this signal? Why don't we represent everything that we have in genomics using these signals? They actually have a lot of other interesting data about the genome itself also that gets lost when you convert it into text, like methylation information, etc., that exists in these signals that could be very useful for downstream analyses, right? That may be useful. I don't quite understand exactly how they could be useful, but many people may be understanding this. But text actually gets rid of a lot of those information. You can potentially actually add them also, but it's more effort also to add them. So our goal is to actually enable both faster analyses as well as more accurate analyses and more useful analyses by converting everything to raw signals. And this is, of course, more sophisticated and even higher cost. Well, higher cost in the sense that more effort, because nobody does this today this way. But we actually have some papers that talk about it. Actually, I'm going to mention some other one later on soon. But I think this is fascinating. If we can actually do this, and it'd be great to get input from people who actually can use this other information that exists in a genome that gets lost in the translation. And also converting. So the reason I actually wanted to talk about this is converting the reference genome from signal to text is actually extremely expensive also. In nanopore sequencing, for example, this in some cases this takes 60% of the time. So it's actually very expensive. So what people use today is deep learning, and this is one of the work that we have done. We actually look into a lot of deep learning methods like long short-term memories, transformers, etc., to do this base. This is called base calling. You call the bases essentially. You look at the signals and call the bases, saying, "Oh, this is an A, this is a C, this is a T, this is a G." That's why it's called base calling. Essentially, today it's done with sophisticated neural networks that are slow. If you can get rid of that translation overhead, you can actually do much better, in our opinion. Okay, if this is interesting, I'm happy to talk more in general also. Okay, let me jump into another direction. Please stop me if you have, or if there are questions, I'm happy to take them right now. Okay, I'm going to go through this relatively quickly, but I will mention something that's also more forward-looking. So today, when you actually do the genomic data processing, you get the data in sensors. It's not a sequencing device. And then it gets stored in storage and then to memory, and then it gets brought into the computing unit. So we're also trying to change this more generally such that you don't need to move the data all the way from sensor storage DRAM to the computing unit, but do the processing on the memory side, on the storage side. Because a lot of genomic data is actually on the storage side. If you can actually offload the processing into a storage device like an SSD, that would be much better. That way, we don't cause a lot of data moments. The general idea is called processing in memory. You basically have, let's say, some important representation of your data. It could be anything, actually. It doesn't need to be genomics also. Basically, the processor says, I want to do this analysis. Do this analysis on your own memory and give me the results. That's the idea. There are many, many questions over here, which I'm not going to bore you with. We've been doing this for some time. And I actually gave a talk at the Academy of Sciences yesterday, specifically on this memory-centric computation. Which is interesting. This changes the paradigm, but it actually makes things a lot more efficient going into the future. But of course, to enable this to be more efficient, because it eliminates the data moment between memory and the processor, or storage and the processor, or sensor and the processor, essentially. So we need to design algorithms to fit processing in memory to enable this. Let me give you an idea of what you can do today with real hardware. This is fascinating, I think. Actually, we found that the sneaky snake algorithm, the state of the art, is heavily bottlenecked by moving data from main memory. And we wanted to perform mapping near where data resides using specialized logic. And so we carefully redesigned the accelerator logic of Sneaky Snake to exploit near memory computation on a real FPGA board. I'm going to give you the picture of this. And if you do this, even in today's systems, you get significant performance and energy improvements, like that much, as you can see. So this is the system. This is a very powerful CPU today, IBM Power 9. It's expensive, but it's bottlenecked by data. So what we do is we offload computation of filtering to this board that has an FPGA in it that has very high bandwidth memory access. So this is a near memory accelerator, if you will. And we study multiple applications, genome analysis one, but we also study another application, weather modeling, which is also clearly very important for climate modeling going into the future. And we see significant speed ups on both, but genome analysis actually have higher speed ups. Essentially, that's the idea. You can offload computation to another device that's near memory that doesn't have to bring the data back and forth to the CPU from memory. Okay, hopefully that gives you an idea. And also there are other technologies that are interesting, like 3D stacking. You can have a logic layer and memory layers over here, and then you can do the computation in the logic layer. I believe these are actually very nicely integratable to a sequencing machine. So the reason I'm talking about this is because these can enable that dream of their device, because these are very, very dense technologies that don't occupy a lot of space in the end. Okay, so let me give you one more thing, and then we're going to jump to the conclusion, if since we don't have a lot of time, I believe. So this is, I think, another part of the future. So one part of the future is an embedded device that can do things. But another part of the future is we have so much genomic data. We want to do huge analyses. We really cannot do these analyses with small sizes of memories. And today in our systems, we have very small memory sizes. But we're talking about petabytes of data as earlier, right? And we may need to analyze petabytes of data at some point. How do we do that? I don't think we can do it by bringing things into the computation units. There's a lot of computation overhead here, and there's a lot of data movement overhead from the storage system. So today we've improved a lot of things clearly. Computation overhead got reduced based on everything that I just discussed. But unfortunately, this data movement overhead still remains. So if you actually reduce some bottleneck, which is computation, the data movement overhead becomes an even bigger bottleneck in terms of percentage, right? So we want to get rid of this bottleneck also. So the idea here, very quickly, is specialize the storage system such that it can do some sort of genomic data filtering quickly. So you build a small accelerator over there such that you send only the data that requires focused analysis based on what we have discussed earlier to the CPU, and everything else stays and gets processed on the storage system in a simple way. This gets rid of a lot of energy inefficiency. And let me give you a number. To process something, to do some simple arithmetic operation is very cheap today. But to move data from storage to computation units, in terms of energy cost, the differential is 10,000x more actually. So moving one piece of data is 10,000x more costly than doing an arithmetic operation on the data. A complex arithmetic operation, actually, not a simple one like I discussed. So this is why we want to get rid of the data movement. This is why we want to actually put everything into the device, because data movement is extremely expensive. So then the question is, of course, what do you filter out here? Exactly matching reads you can quickly figure out. And also based on the principles that we've discussed earlier, if something is not matching, obviously you can also figure that out relatively easily over here. And again, no time to bore you with a lot of these details. It's actually very, very interesting because it also depends on the application in the bioinformatics side, how much benefit you get, because some applications have a lot of exactly matching reads, some applications have a lot of non-matching reads, etc., and the device also. But in the end, if you design this nicely, you get significant performance improvements and energy efficiency improvements according to our prototypes. But we're actually taking this to the next step. We want to actually do this analysis completely inside the storage as much as possible. So if we can actually integrate a lot of bioinformatics analysis inside the storage system, I believe we can actually get closer to this device. Or maybe even if it's not going to be a single device that's operating on petabytes of data yet, it's maybe a much more efficient data center that's actually a single machine, let's say, that's much more efficient than what we have today. Okay, so if you're interested in this direction, maybe a computer scientist or even applications, we do a lot of work in processing in memory. We call this processing in memory, and there are a lot of papers that you can find out. Okay, so I should also say that we have this BioPIM project with a bunch of partners where our goal is to actually accelerate bioinformatics using processing in memory technologies. This is funded by the European Union, and you can see the partners over here. We're very open to collaboration in this domain, including with medical faculty as well, and their courses also. Okay, how much time do we have now? We should wrap up. Okay, okay, sure. I'm going to mention that this is not all, let's say, my imagination. So this is based on decades of work. Finally, major companies are actually building these processing in memory systems. We actually work a lot with the startup. It's a French startup. And we show a lot of benefits on their systems in bioinformatics. But major companies, these are two major manufacturers of DRAM today, main memory. and also Samsung and also Alibaba, they've been building these chips that can do computation inside the memory. And we're actually very interested in accelerating bioinformatics applications with them. I believe there's actually a lot more to do over here. So I'm going to skip a lot of these. But if you're interested in what I just said, we actually have papers that talk about sequence alignment using real processing in memory systems. Okay, this is actually interesting, but we don't have time. Let me go to the final slides. These slides will be available if people are interested. They have pointers to the papers, and some of them have numbers also. So let me talk about future opportunities. Basically, we have newer technologies. This is always interesting. They changed the game significantly. I believe nanopore changed the game significantly, and it reduced the cost so much that you can actually, basically a high school student can buy it and do it themselves. Maybe they're a slightly richer high school student, but they could imagine it, right? And new applications like graph representations are interesting. What I did not talk about is reference genomes are being updated frequently also today. So we don't have a very static reference genome. Going forward, it's going to be much more dynamic. As we figure out reference genome, we want to update it. And maybe you don't want to be constrained to a single, let's say, body updating it. Maybe you want to update it yourself to discover new things, right? So this variations are getting updated in reference genome. And this actually poses even more computational challenges that we've been looking at. I think there's more to do over here. And raw signal analysis. Okay, this is what I mentioned. I think this is a new frontier, and I think this is going to be more important, but we're at baby steps over here. If you're interested in this raw signal analysis, it's a computational paper that is going to be presented in ISMB, International Symposium on Molecular Biology, and etc. over here, as you can see. We're very open to feedback over here, and our code is all available online also. So we would like to understand where we can take this raw signal analysis going forward, both in terms of making the life of a doctor or a scientist easier or accurate, more accurate, and also faster as much as possible. Okay, so let me mention the things happening in industry. I'm going to conclude. This is the platform that I mentioned from Illumina. They have this Dragon platform, very similar to what we have proposed with an FPGA. Again, you can find some information about it, but if you probably talk to them, they can give you more information. I know that some people are actually programming these because they have deals, let's say, with Illumina, so that you can actually customize this yourself with some conditions, of course, with some agreement with Illumina. So this is part of what we have envisioned. And it's great to see this happening actually in real systems today. And you can see this is their claim. We have not tested it, so this is Illumina's claim. We'll see. And there's also GPUs that are actually accelerating things. NVIDIA actually has been putting a lot of efforts. They bought the startup Parabricks that's essentially doing genome analysis, and they're kind of integrating them. And then they've also been adding dynamic programming instructions. So a GPU adding dynamic programming to accelerate sequence alignments is also very useful. But of course, you need to take advantage of it. And then there are a bunch of other companies that do a lot of interesting things that I'm not going to talk about. But let me go back to our dream in the past, right? Our dream was this, right? We wanted this embedded device. Where are we today after 15 plus years? We're not there yet. I believe we still have, we did, well, we and collectively the community did a lot to accelerate things, but we still have a long ways to go. 1 minute is a very high bar. I think we accelerated things like in terms of 100x, 1000x, etc. And we need to do it end to end because there are a lot of bottlenecks in the genome analysis pipeline. If you accelerate read mapping by 1000x, that's great. But then there's some other bottleneck that pops up after that point, and you need to accelerate that, right? So if you want to get to 1 minute, you need to have a very methodical approach in the entire genome analysis pipeline. And then there's energy efficiency, performance. We didn't even talk about security and privacy in this talk, but that's except for the Henrietta Lacks genome mention that I had. But those are actually hugely important issues also. Can you actually do this? I think an embedded device is nice because you can do this securely and privately alone, potentially, right? You don't need to ship your data to a data center that you may not trust. That's the nice part. Or a doctor, a trusted doctor, you can give this device to them and they can do this alone. So basically, we have a huge memory bottleneck also that we need to tackle. Okay, this is the concluding slide. Hopefully, this was interesting. We covered various ideas in accelerating read mapping. I believe many future opportunities exist, especially with new sequencing technologies and new applications and use cases. So going back to the picture that I don't have over here, there's a bright future for intelligent genome analysis. But a very interesting part is really understanding what the devices are capable of and what the algorithms are capable of and what the use cases are so that we can optimize across the stack. Okay, let me, yeah, I should also say that the work that's done that I presented is not my own work in many cases. It's my students and my group's work. And if you want to see some pictures of what they look like, this is what they look like. They all have their own DNAs, if you will. And if you want to learn more about what else they do, there are some newsletters that we put up. And we actually put up the latest newsletter today online. That's why it's not in the slides over here. Okay, all of the tools, especially in bioinformatics, we try to make everything available online. So if you actually use some of these tools, we're happy to get feedback. We're happy to work with you to make the tools work also. And we actually get a lot of requests online on our GitHub saying, oh, this doesn't work. Can you fix it? And it's usually a very small thing that we can fix. It may take you 10 days to fix. It may take us 1 minute to fix. Let me put it that way, because we already have the accelerated analysis that we are done in our tools, right? Okay, so thank you. I'm happy to take any questions. Yes.
First time sequencing genome that hasn't been sequenced before, like I suppose when you put it in, you get reasons many times you put it in, you get reads of different lines, and then somehow you are supposed to use that to reason the genome. But how would that work?
Yeah, so basically, you don't do it completely computationally. You also go to a wet lab. And the wet lab actually figures out the genome in a much longer process, essentially. So essentially, purely computational assembly. So you're talking about assembly. This is called genome assembly. Purely computational assembly is not accurate enough today. We don't have the algorithms to do that. Usually, what people try to do is something called de novo assembly. They construct a graph, and they try to map it to some other reference genome that they may know of, and they try to get an idea of which family it's close to, for example, and they try to refine it. But without really like chemical, physical information that you obtain from a wet lab, it's very hard to validate purely computationally. Does that make sense? So you really need to, that's how human genome was constructed, for example. It was not purely computational. Computation was part of it. Increasingly, computation becoming a more important part of it. Initially, wet lab studies were the real studies that you needed to do to identify these bases, for example. Over time, once you have somewhat of a reference genome, computation starts helping you a lot more. But purely computational construction of an unknown reference genome, I think we're not there yet today. We don't know how to do that algorithmically. You're welcome. That's a great question. That's actually a tough thing to do, I think. Mm-hmm. Yes.
But if you have a small segment of the whole genome, how do you decide where is this number 1 or 10 billion?
Yeah, I see. So yeah, assuming are you assuming that you already have a reference genome or you don't have a reference genome? Okay. Sure. Yeah, if you have a reference genome, it's easy. You can do it computationally. You don't need a wet lab in that particular case, unless you really want to, let's say, validate 100% accurately. But wet lab is expensive, as we all know. So basically, you get a fragment, let's say Switzerland, and then you index into the reference genome. And the reference genome is represented in some way such that when you index into the reference genome, there's some structure that tells you, oh, Switzerland may be matching in these particular locations. There are multiple different methods to do this. One method is called minimizers and seeding and chaining. I don't want to bore you with these details. These are very computer sciencey algorithmic things. But people have developed methods to index into the reference genome given a string. You don't use the entire string also, you use a substring. And basically this index tells you that, oh, these possible places in the reference genome may potentially match Netherlands or Switzerland, whatever you're indexing with. And you should probably compare them. That's the idea, basically. And you get the starting point over here. And if there's an error in the starting point, then that's why we actually do the shifting that I mentioned earlier. That way you can tolerate errors in the starting points that are within these edit distance thresholds. So if you have a reference genome, a lot of things can be done computationally relatively easily. But if you don't have the reference genome, absolutely. What you said is necessary. You need to figure out where these pieces are actually in the real genome. That requires extensive wet lab analyses. Yes.
Can the filtering of the genome be the AI version of pre-processing the text within the machine learning, for example?
Yeah, certainly. I think there are similarities. So what we have done with filtering is heuristic-based methods, but one can actually use machine learning-based methods also. Now, I should also say that I think it should probably be a combination, because with some heuristic-based methods that are extremely fast, so the advantage of some of these heuristics is they're extremely fast. Machine learning, if you go through a neural network, for example, it's usually expensive to do that. So I think there is a place for machine learning, but we need to use it carefully. So, I mean, a lot of these issues we tackle actually in that read mapping, 111 pages. But there are different approaches to this. Usually indexing and filtering can be combined actually in some way. There are different approaches, of course. You could do one earlier and the other next, but in existing systems, usually they're combined. But that's a very good question. Maybe there's another way of doing this, right? Yeah. Oh. Okay. Okay. Yeah. So this is another area that I do a lot of work in. In fact, the earlier talk that I gave at at the University of Montenegro was about issues with these memories. So I think what you're saying is, what is the effect of this new RAM on, let's say, an application like genome analysis, right? I would say that not much. So the benefit is you get higher capacity with these new. So in that sense, it's good. But in terms of the speed, like what you're mentioning, you're not going to get a huge difference. And we actually did a lot of benchmarking. We've been doing work in this memory area for a long time. I can point you to papers that show that some new technologies are actually slower than old technologies because of some of the trade-offs that are made in the design. Usually the design is optimized for capacity, meaning with each new memory technology generation, you get higher capacity devices, which is great, of course, for storing more data. But this comes at the cost of additional latencies, unfortunately. Yeah. Yes, please, another one.
Genome for the first time. If I have a genome and I put it in the machine, and for example, the first time I get one sequence of 9 letters and one sequence of 9 letters, and then the next time I put it through, I get a sequence of 15 letters. And if I notice in the first sequence is that the 3 letters of this one and the first 3 letters of this one are like this, like they are matching, and I want to suppose that it's that it's where the genome connects, and the next genome from the second time I put it in, the 51 chain, is also matching to that. Yeah, I suppose, and that's the correct order of the genome. Why cannot we do that on a bigger scale and teach the machine to do that for us? That way, just finish the sequencing.
So you're talking about genome assembly again, right? First time. Yeah, I think essentially this is so very, very good. It's good that I don't know if you know about this field before, but you're developing some of the ideas that have been developed in the field. What you're saying is essentially it's called read-to-read overlap finding. You're finding read-to-read overlaps, and actually that's one way of doing genome assembly using computational methods. So by finding these overlaps and stitching them together, you can actually find something. But can you be sure? That's the difficulty. You cannot be sure completely. That's why you need the wet lab. Does that make sense? We can discuss it more, but feel free to ask another one. Yeah.
We mentioned that we sometimes do sequences like 30 times.
20 times, yes.
Before the 30 times, 60 times.
Okay, good. Exactly, yes. Exactly. So that's essentially what is. So now I think you're building some de novo assembly techniques. This is called de novo assembly, and essentially it's done that way, sequencing many, many times, trying to build confidence. Even then, it's not that easy. I'm happy to talk more. Maybe you'll find some breakthrough. Who knows? But if you're interested in genome assembly, this is a very interesting, fascinating subject, and certainly you can read more. But in the end, you cannot be sure. Basically, in the end, you need the wet lab to construct this thing and say, okay, what I've constructed is actually good. You may build more confidence, but with the error rates that we have, it may not be enough. And also, you get variations, right? Well, I guess with a single genome, you don't have variations, but okay. But errors can actually compound as well. Okay, you're welcome. But read-through overlap finding is actually an area that also within itself is computationally costly. We analyze it in some of our papers. Yeah.
Errors, are there, like, have you noticed that there's a specific line of error that happens in machines? Can you maybe try, I mean, probably there is, because like 2 days it may have something similar and that machine may work and look exactly similar, and that may mix them up. Like, are there some specific things that happen always, or is it different for each and every machine?
Yeah, so that's a great question also. So there are 2 types of errors. First of all, it's different across classes of machines. Like nanopore has a very different process. I didn't talk about the machine technology. I think I have some backup slides that talk about machine technology. We can go into them if you want. But nanopore has a very different method. The graphene nanopore, you pass the DNA strand through it, and then you measure current. Whereas Illumina, essentially, you chop the DNA into very small fragments. You send it into a light sensor. That's why it's called Illumina. And then this light sensor gives a different reaction, different refraction, essentially. And based on that refraction, that you figure out whether it's an ACTG. So the different mechanisms are clearly different in those machines. But within a family, let's say nanopore, let's pick one particular nanopore, there are errors that are systematic across the nanopore device based on the construction of the device itself, because it's a graphene nanopore, and graphene nanopore makes mistakes in terms of substitutions, for example. They have, so some devices have more substitution errors. Substitution is mismatches, right? You get an error and you think it's a mismatch, but it's actually a machine error. Machine somehow caused this mismatch, right? Because of the way it senses the current. So certainly different devices have these different systematic problems. And if you know exactly how the device operates, and if you can analyze it, you can correct for it. And you can minimize the errors. And nanopore sequencing initially had error rates around 75, 80, 85%. Today we're at 90, 92, 93%. And some of that improvement happened because they understood the machine and they got rid of the systematic errors as much as possible. Now there's also a device-to-device variation. I have a nanopore device, you have a nanopore device. These are variations, and they may actually give different errors also. Yeah, this is a fascinating area, but it's a bit harder to do research in this area because you really need to get into the guts of the device. So Oxford Nanopore Technologies, they should be the ones who are doing this research, and I think they're doing a good job in improving their own machines. But of course, there's a possibility that you can also understand this if you look at the raw signals that are generated and try to figure out, let's say, some inaccuracies in the raw signals. Yes, there's another question over here. So whoever, you have the priority, I think.
Problem is an optimal length of the reads, or it depends on the game, on what we're studying, or it depends on the machine it's analyzing.
Yeah. So basically, it depends on machine. Essentially, you don't have control over the length of the reads. You're at the mercy of the sequencing device. Even the sequencing devices have different lengths, so they don't generate perfectly. So, for example, nanopore can generate reads of length 50,000 to 2 million base pairs. And you need to map them basically once you get them. So you have no control over it, as far as I know. Yeah. Yeah. Yeah, I think so. I mean, certainly you can do that sorting, but in order to be able to do that sorting, I think you need to somehow reconstruct what you're getting out of the machine, right? Because what you're getting out of the machine, let's say it's 1,000 base pairs, it's not enough for you to know where it's coming from in the reference genome, right?
Okay, I see. I mean, like, we have some special regions in between.
Sure. Yeah. Okay, so basically you have the particular sequence of characters that are you know are okay. I see. Yeah, certainly. I think some of these analyses can be done without fully mapping all of the reads that you get, and you look at your reads quickly and okay, these are the interesting ones that I want to do. And this is already, well, I shouldn't say this is already done generally, but this is something that also aligned with what we were discussing, right? Eliminate parts that are not interesting or they're not useful. But yes, if you know enough information about the specific analyses or specific parts of the genome and specific characteristics of those parts of the genome, you can guide the read mapping tool to do that. Unfortunately, I'm not aware of read mapping tools that are easily configurable to do that. You may need to go and modify the tool itself to do it, but I know how to do that. Basically, it's not that difficult, but I don't think a lot of people do that in the end. Yeah. Yes, one more question here. I think there are two. Yeah.
The shifted having this, is it possible that it can classify relevant sequences as too dissimilar and therefore irrelevant? Also, what is the case if there are multiple deletions and in between those there are some mismatches? How does it deal with those?
Okay, let me take the first one and then try to understand the second one, because I lost my train of thought on the second one. The first one, basically, is it possible that it can say two sequences are too dissimilar even though they are similar? Yeah, basically, with shifted Hamming distance, no. Basically, we have a mechanism and a proof in the paper that shows that you're never going to say two sequences are dissimilar if they're similar within a threshold, right? And you can read the paper for more detail, but basically you need to be conservative in what you eliminate. And once you're conservative enough, you don't eliminate sequences that are potentially similar. We call this, like, let's say, you don't eliminate things, useful things, basically, and you don't want to eliminate those useful things. And it's very important for these filters not to eliminate useful things. What was your second question?
Mismatches in between all deletions. Does it basically shifts them, but does it identify that something is a mismatch, or does it throw it away as then?
Okay, so that's an excellent question. So the purpose of shifted Hamming distance is not to exactly identify mismatches, deletions. It's to the purpose is to filter out things quickly that are not going to match. So we don't care if it's a mismatch or a substitution or a deletion. If it's going to be dissimilar enough, throw it away. Yeah, very good question though. Later works if you try to make shifted Hamming distance identify things, then you start becoming closer to edit distance computation. I need to use some other algorithm. Sure. What? Yes.
So when they're sequencing and turning the genome into letters, I suppose that the computer needs to go like either two ones or two zeros or multiple of it. But like when it comes to quantum computers, I don't know whether you're familiar with them. I suppose you are. Like they have 4 options instead of just 2. So is it possible to use that to minimize the memory needed for genome sequencing?
So you said quantum computers, right? Okay, I just want to make sure that I heard correctly. Basically the answer is I don't know. There's not much work in this area. Quantum computing is very, let's say, fresh field right now. I'm very interested in the implications, but I'm not sure yet. I'm not sure if it will actually help with the memory size, but it could potentially help. What helps with the memory size is compression. So a lot of these reference genomes can be compressed, and people actually do that, and you can operate on compressed data. This is something we're also working on. But once you go to quantum, a lot of things change. I think this is an area where there needs to be more brainstorming, if you will. Does that make sense? Or? Yeah. Yeah, I mean, I'm happy to brainstorm. This is a bit forward-looking. And I'm actually quite interested in implications of quantum computers in more difficult analyses like this. Maybe there's something there, but it requires some, let's say, thinking of how to change the representation of the reference genome as well as any genome, and how to actually change the algorithms to actually operate using quantum computing principles. That's why I cannot have a good answer to you, because I have not done enough thinking on this topic, but I think it's fascinating potentially. Yes, one more. Mm.
And if my understanding is that the major advantages are, number one, well, by moving into the text, we basically lose a number of important glitches in the signal domain, which may contain a lot of hidden knowledge.
Mm.
And number two, well, if you do signal processing, then you have a wide plethora of methodologies already available from phase analysis or earthquake detection, etc., etc. That you produce with minor or major adaptations.
Mm.
Maybe a number three would be something that doesn't come to my mind, but you see how maybe like to study.
Yeah.
So what are the major advantages here?
So I would say you nailed both of the advantages that I have in mind. I don't have a number three, but maybe somebody else here has a number three. But basically, I think these two pieces are the major advantages. If you just look at the raw signal, you have information there that gets lost when you translate it to text, like methylation information that we've discussed briefly, and other types of information that could be there, hidden or not hidden. Let's say both of them. A lot of these are actually not hidden, but they just get ignored when you go to text. But there could be hidden information in the signals also that we don't quite know. So there's that information that could be useful. And then the second thing, maybe I would refine the second thing a little bit more. Or maybe I'll add a third thing. Okay, the second thing I think is, what you said second is actually different from what I was thinking about, or maybe I was combining both of them. The second thing you said is you have signal processing, now you have a signal. Now you can operate on the signals using these signal processing techniques we know about from other domains, right? Including time series analysis, which is used in many domains, for example, including astronomy, economics. Basically, there are a lot of things that we operate on a lot of analog signals today. I believe we still need to specialize our analysis. I don't think we're going to basically take something from some other domain, but maybe we'll need to specialize it. So I think that's another advantage. But the third advantage is probably different from the second one, which is we get rid of the inefficiency of conversion. There's an inefficiency of conversion from raw signal to text. And that's actually quite inefficient. And if you just operate on the raw signals, you get rid of the inefficiency of conversion. Of course, you're going to replace it with some of those techniques. So that's why I think these are coupled with the second one you said. But maybe that inefficiency of conversion is huge to begin with, right? It's actually very large. Okay.
If you see a single character level difference between the human genome generated controlled by the Institute in Boston, and what you currently have, you don't know if this difference is a consequence of a mutation or of some impurity in the chemical process. Well, with this approach, I think you drastically increase the probability that the difference is not due to impurities in the chemical process, but due to computation.
Mm.
Am I correct?
I'm not sure about that because I'm not sure if I fully understand it. Because what you with raw signals are the signals that are generated by the nanopore device, for example. So it has the signal itself is measured by the nanopore device. So it has all the impurities, all of the chemical processes. So it's contained in the signal itself.
To decide about ACGT.
Yeah. Uh-huh. Yeah. Yeah. That, I mean, that's the hope basically. But yes, I agree. You can apply techniques to reduce the errors and identify them potentially. That's being done today using some post-processing techniques like machine learning today. But yes, I think we need to look into more signal processing as well. Yeah. Mhm. Yeah. Yeah, we can talk more. One more question.
And like each base would have its own wave in the time. And then when you have a sequence with 4 letters, you could use like adding up those waves, or I don't know, adding up those waves, and then you would get one final wave. And instead of just doing the alignment usual way, you could maybe compare the waves and see whether the sequence is similar. Would that be possible at all?
I believe so. I think these are some of the things that are enabled by looking at the signals. And you can convert them to the wave domain, right? Using wavelets, for example, and then do the analysis that way. So I think it's possible, but we need to investigate that. Those are some things that we're very interested in looking into. This is a very, very pure area right now. No one is looking into it as far as no one is a very, let's say, overstatement, but no one has, let's say, published works like that you mentioned. Yes, please.
Thank you for this insightful lecture, and congratulating such huge achievements being made in accelerating these processes. And I would just like to say I really fully with Professor information about the revolutionary way you analyze interesting data, but to the core molecular biology question remains to analyze by radical sequencing with the level of the signal, how can we translate it to something that is understandable?
That's a great question. So certainly at some point the translation needs to happen, right? But hopefully most of your analyses on the signal domain, meaning you represent the reference genome using signals. Of course, this is going to make the analyses also more potentially storage. It becomes huge, for example. So let me give you some numbers over here. So I'm actually kind of contradicting myself using this real signal analysis, but that's why it's challenging. Okay, let's look at this numbers. For example, this nanopore device, right? Raw signals are huge. This is the base calling step, basically convert raw signals to text. This is actually taking a whole lot of time, basically. It reduces the data set size, as you can see. So every analysis, if we get rid of this base calling to reads step over here, now you do the matching of the reference genome, everything using raw signals. At some point, I think you'll need to do the conversion. But hopefully by the time you need to do the conversion, you did a lot of analysis that was efficient with methods that are to be developed. And once you do that conversion, you're going to do the conversion on a very small piece, maybe one part of the reference genome that is maybe 40 signals, and you want a single base from there, right? So yeah, eventually, so we postpone the space calling somewhere else at the end, hopefully. That makes sense, hopefully. I mean, alternatively, of course, everything, even the humans start thinking about signals, but that's a tough task.
Yeah.
Yeah,
Sure.
How far we are from the time that we can really put something like such a device, some future device probably, in a huge treatment plant and measure, let's say, the state of the population, health population. It means that is it too much information that we never can reach, or it's possible somehow? You see that it's possible that we can go so far that we can get so much information and make some analysis and say, let's say that we have some pathogens in the city or something like that.
Yeah.
Is it possible with this technology? Can you see this future, or it's just beginnings? That,
Yeah. So I missed one part. Where do you want to put this device? In a
huge, let's say in the end where.
Oh, okay, I see. Okay, something. Okay. Oh, okay. Yeah. So I think that's a great question. Well, sure. That's a good place to analyze for sure. I mean, COVID-19 benefited from that analysis quite a bit. Yeah, sure. There's a lot of information. So, okay, that's I mean, it's I believe it's possible. I mean, I can imagine a future where that's possible. Actually, it's not, in my opinion, depending on. Yeah, exactly. Yeah.
But just for one virus.
Exactly. Yeah. If it's lots of things, everything you want to detect. Yeah, it depends on the scale of the analysis, but I think it's possible. I don't see any fundamental problem with that. It's
because it's a huge base for information that can be exchanged between the populations.
Yeah.
If there was some AI in the future, can analyze this to get all information.
Yeah, I think so. I mean, initially, like in the shorter term, that machine may collect data and send it somewhere so that the analysis is done somewhat close by, let's say, in the wastewater plant itself. But in the overall, very like longer term future, I can see with some of these extremely efficient techniques that are the future versions, let's say, of these, you can actually discard a lot of useless data, but focus on the data that's actually interesting and basically send messages to people saying, oh, you should be focusing on this data that I'm sending you, right? So I think it's not that far into the future, in my opinion. And it's very useful. That's another application, I think, of this, let's say, vision. I mean, that's a great question. Maybe the right answer is I don't know everything about Switzerland, but I don't think it's incorporated enough, in my opinion. Yeah, there's some, and also it's still costly. There's some, I think there's some level when a doctor requests it, you can get it done, but it's extra cost in the end. But it's not, I think we want to take it to a level where you go to the doctor quickly figures out what you're vulnerable to. And even before like talking to you at all, they already know a lot about you, let's say. And maybe not just the doctor, the device itself does this at some point, right? So we're definitely very far from there. I think we're still very like baby steps. Maybe there's another question in the back. So that was not the last question. Yes. Okay, sure. I mean, I can elaborate a lot more if and when I come during that time. I would hope to, of course. I would hope that if is eliminated and it becomes when. But I think this is an interesting area because CRISPR or gene editing technology has been very useful in understanding certainly the genomes and also other potential things like fixing the genome going into the future or fixing issues, right? we're looking into it from, again, a computational perspective, and we want to really understand what are the bottlenecks in this technology. Like, what is similar to what we have done over here, right? What is really bottlenecking people so that this technology can be used very quickly to make quick analyses? Because if every time it takes a very long time to do the analysis, can we actually accelerate it somehow so that we can actually much more quickly converge into discovering meaning out of particular genes, as well as potentially editing, if that's going to be interesting. So this is the general direction, essentially, that we have in mind. We have some results, but they're not at the point of exposure yet at this point. Hopefully, by the time that diaspora happens, then we will have something published, but we'll try. Yeah, one more question. Okay, we can play the game of who's gonna ask the last question, and we'll be here forever. No, it's okay. I didn't mean to put you on the spot. It's fine for me. Sure.
How does this machine get these irrelevancies? What exactly are they?
So what do you mean by irrelevant sequences? Can you tell me?
So, can you speaking before shifting in the override in the IEDM slide?
Okay, let me go over there. Well, oh, I see what you mean. Basically,
what are these irrelevant sequences for this whole process that we need to eliminate so we can get a lower computation power for the rest of the.
Yeah, so I think that was a very general slide. Intentionally, that was very general because it covers a lot of different things. It depends on what you're looking at. Okay, this is the one I think that you're talking about, right? Yeah. And what this is doing here, but what's okay. So basically, I wouldn't call it irrelevant sequence. I would say you have a sequenced read, and this part is not matching a particular portion of the reference, right? It could be matching some other portion of the reference, but it's not matching this portion of the reference. So do not do expensive computation on that. So in that case, the sequence itself, the read itself, in that particular case, the sequence that you got, the read that you got is relevant, but the part that you're examining is irrelevant for that particular read. You should really focus on where it's matching. That's one case. But the slide is also general in the sense that there may be cases where the sequence that you are looking at is completely irrelevant to the sequence you're matching. For example, in the metagenomic analysis that we said that we looked at, right? You have a thing you have sequenced, and you're comparing to thousands of reference genomes to figure out where it's coming from. Maybe none of them will match, right? If you can quickly figure that out because of, again, the same principles, you can get rid of a whole lot of computation, essentially. So the irrelevance depends on the use case as well as the local, the general use case, as well as where you're looking or where you should be looking in the reference genome.
It's just matching a different type of reference that we are currently.
Exactly, yeah. That could. Yes, one more.
When I hear genome editing, the first thing that comes to my mind is that when you take somebody's genome to figure out the mutations that make a person prone to some illnesses inherited from ancestors, then you make the proper changes, you return them into the body, and that person becomes, quote unquote, a better person. Is my understanding correct, or I was just misled by the syntax?
I mean, I believe maybe there are other people who are more, let's say, experienced in this who can speak to that. I mean, that's certainly one, let's say, futuristic thinking of that. Yeah.
When he's just one cell or 2 cells, from the beginning.
Exactly.
Yes, we have it. Yeah, we all agree. It's with some animals.
Animals, yeah, not humans yet.
combination. That's here in Montenegro. No, in Montenegro. It's bring the role in that state. You don't be in Montenegro because the second one that comes to my mind is that we all become ideal. Then, well, we may keep killing each other. You remember the tragic case in Belgrade? The kid was killing only the other kids that scored better in the school.
It depends on the definition of what ideal was.
Right? Or if that is something, it's not the J, what is that? It's not just about J. It's about all combination of everything. It can be a methylation of some genes too. It can be a technological solution. So we cannot.
Somebody has more questions, maybe we can come to talk a little bit more, but I would like to thank our speaker for excellent presentation. It was very informative, and I think everybody learned something from it. And thank you for answering questions. I hope you will be able to come in again.
I hope so too. If not, some other time, of course, for a longer time, hopefully. But thank you for hosting me. And thank you for the lively, many questions. Feel free to, if you ask more questions, feel free to email me, or if I have time right now, I'm happy to talk also. Thank you.
Yes.